Hello, everybody. First, I would like to thank the organization committee for this opportunity to share our work. My name is Riyue Bao. I am Associate Professor from the University of Pittsburgh and UPMC Human Cancer Center. Today, I will talk about molecular correlates and therapeutic targets in T-cell inflamed versus non-T-cell inflamed tumors across cancer types. Immune therapy has emerged as one of the major treatment for patients with cancer. However, less than 50% of the patients respond. Therefore, the overarching question that we are interested in is why some patients respond and some do not. Our working model defines two phenotypes: the T-cell inflamed tumors, where the immune molecules such as CD8 and FOXP3, as well as the immune checkpoint molecules such as PD-L1 and IDO1 all light up, and non-T-cell inflamed tumors, where none of those molecules are present. In clinic, patients with inflamed tumors are more likely to respond, and those with non-inflamed phenotype do not. Therefore, the two biological phenotypes can serve as surrogates to study the molecular mechanism that drive response or resistance. Previous work from our group and others has defined an interferon gamma associated signature from gene expression profile to model such phenotype. In melanoma immunotherapy trials, higher interferon gamma signature score predicts better outcome. Using this working model, we sought to make an unbiased discovery of molecular correlates associated with T-cell inflamed or non-T-cell inflamed phenotype across 31 histologies from TCGA, the Cancer Genome Atlas. We first categorized patients into non-T-cell inflamed and T-cell inflamed groups based on a defined T-cell inflamed gene expression score. Then we contrasted these two groups to identify mutations or pathways enriched in each phenotype as potential candidates for further studies. In a pan-cancer analysis consisting of more than 9,000 tumors, we discovered 30 genes significantly more frequently mutated in the non-T-cell inflamed tumors and 51 genes associated with inflamed tumors. For the interest of time, I will focus on non-T-cell inflamed tumors. The top head, as you can see, IDH1, is predominantly mutated in glioma, a tumor type that does not respond to immune therapy. We found that IDH1 mutations are enriched in non-T-cell inflamed relative to inflamed phenotype. And based on this data, as well as other experimental evidence, we have launched an EVO plus NIVO therapy in IDH1 mutant glioma and advanced solid tumors. This trial is led by Dr. Jason Luke, clinical PI of our lab. However, most of these mutations occur only in a small fraction of tumors. Therefore, we started to ask whether there are other mechanisms that could potentially drive immune exclusion, if not mutations. To address this question, we performed a causal network of pathway activation analysis and identified pathways that are significantly activated in non-T-cell inflamed tumors relative to inflamed tumors. 31 pathways activated in 4 or more tumor types are shown on the left side. In particular, I would like to point out that those pathways highly interact with each other, as shown on the right side. We also found a greater number of activated pathways a tumor has, the more likely a tumor will have a non-T-cell inflamed phenotype. Taken together, we believe it is reasonable to speculate that maybe the tumor-resistant pathways crosstalk with each other, and inhibiting one pathway might not be sufficient to convert cold tumor to hot tumor for improved response. Therefore, rationalized co-inhibitory or co-stimulatory therapy plus checkpoint inhibitors should be prioritized. Lastly, I would like to show you that we have taken these pathways and performed extensive validation. One example is KLF4 in anti-PD-1 treated patients with metastatic melanoma. KLF4 is a key player of sonic hedgehog signaling, and its activation score is significantly higher in non-responders compared to responders. Based on our finding and other experimental evidence, we have proposed a new clinical trial on smoothened inhibitors plus anti-PD-1/PD-L1 in tumor types that harbor sonic hedgehog activation. In summary, I hope we have convinced you that we identified oncogenic mutations and activated pathways significantly associated with non-T-cell inflamed or inflamed phenotype. We also were able to validate these findings using protein array data and also in independent external cancer genomic datasets, including ICGC, CPTAC, and MET500. Data were not shown here due to interest of time, and we have mined in existing drug database to nominate molecular targets and drugs potentially available for immediate translation into clinical trials for patients with cancer. With that, I would like to thank members of our lab, especially Dr. Jason Luke, clinical PI of our lab, and Dr. Daniel Staper, who also really helped drill down some of the candidate genes. This work was partially supported by Human Cancer Center funding, DoD, and NIH CCSG grants. If you have any questions, please feel free to reach out to me or find me on Twitter. You can see my contact here. Thank you very much, and I hope you enjoy the rest of the conference.